Hello, medical intervention students. I'm just going to give the instructions for the outside of class work to be done for Tuesday, December 1st and Wednesday, December 2nd, 2020 for the time that you are not in class. Okay. So if you're on blue, this is what you'll do on gold and vice versa. So when you are in person, you will be going through the ELISA procedure, like we talked about, to actually go through those steps. So as I outlined today in the November 30th lesson, it's important that you read through that in advance. But we're going to go through those steps and try to get through as much of that as we possibly can tomorrow, which today's Monday, so that'd be on Tuesday. So whatever day you are present, Tuesday or Wednesday, we'll work on getting as much of this done as possible. Additionally, your outbreak analysis is something that you should be working on today. That's the new assignment. Everything else should be caught up to this point. All the other assignments, if you're missing things, make sure you get those in as soon as possible. Can't tell you how important it is that you watch these videos. We'll not be having Zoom calls going forward as when we're in the hybrid, so there will not be Zoom calls. So it is important that you watch these videos, especially if you are a gold student and you're coming in, and that way you don't lose a day, so you can get the stuff done in advance. I'm going to make sure I have it in place to where you can get the things done in advance without being in person. The things we're going to do in person are going to be more hands-on lab activities, and so along with some instruction as well, and I'm always able to help clarify things. So you'll see this video posted here. It'll say December 1st and 2nd, 2020, and so on. So there'll be a video for every 2 days to show you what you need to do outside of class. So we'll be doing that ELISA procedure. You should be working on this already. And then we're going to start the 1.2 section. And 1.2, we're finally out of 1.1, and we kind of know what all the people have, and we'll go over those results in class when you're in person. But 1.2 is about antibiotic treatment, okay? And so 1.2.1 is antibiotic therapy. And so really you're looking at how antibiotics are treated, because we know Sue Smith has this Neisseria meningitis bacterial infection, and we're going to try to treat that and cure it. And so how do antibiotics work? Well, in order to understand that, you need to know this basic structure of bacteria, and then also different types of antibiotics and how they work. So this assignment is not due until the end of the week, until December 4th at 11:59 p.m. So all the way through Friday. There's going to be new work that you'll be assigned, though, Thursday and Friday. So there's going to be kind of a lagging due date on things, but you definitely don't want to put things off and get really far behind. So you'll see this diagram here of a bacterial cell, and you will be getting a paper copy of this assignment in class. So there is a paper copy, so you'll get this on paper if you want to do it. If you're at home and you're not here on Tuesday, you can definitely get started on stuff and leave some room for you to do the drawings later, but you can definitely type things in as well. So you are going to turn things in digitally still. You're not going to turn things in by paper. You're going to turn things in online. But that paper copy is there in case you want to handwrite some of the information. So with that, and usually just take pictures of what you complete. Here's the 121 workbook. It is located here in Canvas. 121 workbook. You bring this up, and so you're going to do you're going to draw the cell, and make sure you color code each structure and add a legend for each corresponding term and describe each part next to the term. So there's quite a bit of stuff here to cover. There's this bacterial structure video that helps you to learn more about that. So make sure you watch that too to help you. Okay. And so here you'll see those structures below. So instead of writing it next to it, make the drawing here. And then if you want to color code each of these, that's fine. Or make a color code of your own. But then you can just type their functions in about what they do below, or description. Then gram positive versus gram negative bacteria. If you go back into PLTW, all right. It talks about gram positive versus gram negative bacteria. Now we're going to determine if it is gram positive or gram negative. So we're going to look at the difference between those. Figure 1 shows the difference. If you look here, gram negative bacteria, if you look at their cell walls here, okay, and you get gram positive bacteria, see this blue layer is really, really thick. That layer is called peptidoglycan. And peptidoglycan is a carbohydrate that's a really thick layer on gram-positive bacteria. Gram-negative bacteria, you see, is really this red layer is really, really thin, and that's a big difference between them. When you think about antibiotics, they're trying to kill these things, so you got to find what you can target. And if you have a thick cell membrane versus a thin cell membrane, that might make targeting a little bit different in how that medicine is going to work. All right. And so if you look at this, you see a comparison. Some of it's filled in for you. You do not need to do the in-text citation on these. So just heads up. Don't worry about doing that. But you see gram-positive, cell wall, and peptidoglycan. Draw and describe. Stain results in coloration. So, for instance, final color is pink. All right. For these, whereas this would be different, right? You'll see the look up the information here. Does it have an outer membrane or not? Toxins. Does it have toxins or not? How does it respond to antibiotics? What are some examples? All right. Other info. Maybe there are some diseases that are more linked to that than others. And then finally, classes of antibiotics. And so here you can see the 4 types: penicillins, tetracyclines, fluoroquinolones, and sulfa antibiotics or sulfonamides. It's known as sulfa drugs. And so those, you're going to do some research on each of those, and basically look at where and how it affects the bacterial cell. So how does it work? What types of things does it work on? So you can see examples, names. How does it work? For instance, tetracyclines prevent the growth and reproduction by preventing the translation of proteins. All right. And then maybe a sketch of it, also a sketch of the mode of action. And finally, is it effective against gram-positive, gram-negative, or both? All right. And then finally, research Neisseria meningitis. And what does it look like? Is it a rod shape, which is a bacillus? Is it circular shape, which is a coccus? Is it spiral shape, which is a spirillum? Is it gram-positive, gram-negative? And how's it treated? So you basically have all week to be working on this as well. So you really have two assignments due that are primarily outside of class work time, and that would be the 116 and the 121 workbook. And then we'll work a little bit more. You'll probably have a little bit of time on this on Thursday, Friday to ask questions, and we'll go through some of that in class together. So, so that's what your primary focus this week. We're going to do the ELISA procedure in class. We'll work on this a little bit in class, summarize this, but primarily we're going to be working on this ELISA procedure so you can actually do it in class. So after you're done with this, you need to be working on the 121 workbook on the Tuesday, Wednesday days. All right? Let me know if you have any questions, and I'll see you soon. Thanks.